First of all, on behalf of the organizers of this Congress, I would like to thank all participants for coming and my co-organizers for the great work. The theme of my presentation dedicated to Drosophila life extension by overexpression of the growth arrest and DNA damage 45 gene. This work was done in our Institute of Biology in Syktyvkar, and this Friday we will celebrate 50 years anniversary of our institution. It is well acceptable that the irreplaceable nature of the DNA sets it apart from other cellular molecules and makes it a critical target for age-related deterioration. This phrase I've got from the paper of Vera Garbunova and Andrei Selyanov, who are classicals in the theme of DNA repair and DNA damage with age, changes with aging. It is well known that many kinds of DNA damages increase with aging, including abasic sites, nitro, oxo, and glyoxal guanosine changes, DNA double-strand breaks, and DNA protein cross-links. And GAT45 proteins are involved into DNA damage with stress response. DNA damage induces stress response pathways which activate the activity of P53 and all FOXO transcription factors which transactivate GAT45 genes expression. These genes are involved into mismatch repair, nucleotide and base excision DNA repair, DNA methylation and gene expression, and depends on the cell types, cell cycle arrest or apoptosis. We recently observed that GAT45 proteins related to aging, longevity, and age-related pathologies. They involved into metabolic damage, aggregates accumulation, oncogenesis, immunosenescence in mammals. The aim of this study to investigate the effects of overexpressing Drosophila GAT45 gene in the nervous system on the lifespan, age-related pathological decline, and stress tolerance. First of all, we studied the spontaneous expression level of GAT45 gene at different ages of wild-type strain continuous. In young flies, middle-aged flies, and old flies. We observed that spontaneous Drosophila GAT45 gene expression level drastically decreased with aging, 10 times in comparing with young flies. That's why we tried to overexpress this gene with the using of GAL4 US Drosophila overexpressing system in the nervous system of fruit flies. We observed about 5 times overexpression in males and about 2 times in females. This constitutive overexpression leads to increasing of median lifespan, more than 70% in males, and more than 20% in females, and increasing in the maximum lifespan, more than 22% in males and more than 10% in females. We use conditional overexpression of the same gene in the nervous system to prove this previous data. We observed 4 times overexpression in males and 2 times overexpression in females. And we revealed the increasing both in maximum and median lifespan in males, and the increasing in median lifespan in females. Then we studied the effect of postponed conditional overexpression of GAT45 gene in the nervous system, and after 30 days of age, we induced overexpression, and we still have the increasing in the median lifespan both in males and females. Sometimes the increasing in the lifespan accompanies with the decreasing in life quality. That's why we studied the fertility and the physical activity of flies with the GAT45 expression. We observed that constitutive expression of Drosophila GAT45 in the nervous system increased female fecundity in different ages, and does not decrease the male fertility. The conditional overexpression does not change female fecundity or male fertility. We applied spontaneous activity test and negative jet-axis assay to compare the effects of constitutive overexpression of Drosophila GAT45 gene on physical activity, locomotor activity, and we observed the increasing of spontaneous activity both in males and females, and negative jet-axis level, as well as the prolongation of flies activity. The conditional overexpression does not change the spontaneous activity both in males and females, but still increase the negative jet-axis level in different ages. What are the reasons of these effects? One proposition is that GAT45 overexpression affects the DNA repair. With the using of Komi assay, we studied the spontaneous DNA damage level, EP sites and DNA single-strand breaks in larva brain. In the case of conditional and constitutive overexpression of Drosophila GAT45, and we observed the decreasing in the level of spontaneous DNA damages level. But the apoptosis frequency does not change. The second proposition is that GAT45 expression affects the stress tolerance. We observed that overexpression, constitutive overexpression of Drosophila GAT45 decreases the mortality level after paraquat treatment, both in males and females. And conditional overexpression decreased the mortality after paraquat treatment in males. Constitutive overexpression of Drosophila GAT45 affects the resistance to heat shock. We observed the decreasing in the level of mortality after heat shock, both in males and females, with overexpression of this gene. Conditional overexpression also affects the resistance to heat shock, both in males and females. We studied the resistance to starvation and the level of mortality in the case of starvation decreased after overexpression of this gene, both in males and females, but not in all cases, just in comparing with one of the control line. The conditional overexpression affects positively the resistance to starvation just in males. We collected some data about the mechanisms of possible mechanisms of GAT45 effects on the aging speed. First of all, GAT45 help the cells to maintain the genomic stability, as well as GAT45 involved into regulation of cell cycle, cellular senescence, and apoptosis. Both of these suppressed the carcinogenesis. GAT45 genes in mammals affect the cell differentiation and the maintenance of a cell pool. That's why it could suppress the atrophic and the degenerative processes. We found the paper about amelioration of chronic insulin resistance due to GAT45 gene. And this gene involved into the suppression of autoimmunity and immunosenescence too. What are the mechanisms of such effects? We observed that GAT45 involved into protein-protein interactions with the cell cycle and DNA replication regulators, mitogen-activated protein kinases, and nuclear hormone receptors. We've done this with the using of bioinformatics approaches from the literature data. It was recently revealed that GAT45 involved into DNA demethylation. That's why it could affect the transcription of particular genes. And GAT45 alpha binds RNA, but not single or double-stranded DNA or methylated DNA in vitro. And GAT45 alpha displays RNA-sensitive colocalization in nuclear speckles with the RNA helicase P68 and RNA binding protein SC35. To summarize, we observed a 10 times decrease in spontaneous activity of Drosophila GAT45 gene in the nervous system of old flies of wild-type strain Cantonese compared with young flies. Demonstrated significant increase of median and maximum lifespan in both males and females with the constitutive conditional and postponed conditional expression of this gene in the nervous system. The most obvious increase was observed in the case of overexpression of GAT45 at early life stages. The lifespan extension effect is more pronounced in males than in females, in accordance with the sex-dependent overexpression of this gene. Constitutive and conditional overexpression of Drosophila GAT45 in the nervous system increased or did not affect female fecundity and male fertility, as well as nervous and muscular activity. In the neuroblast of Drosophila 13 star larva with the constitutive and conditional overexpression, DNA damage level is reduced. Hence, the longevity with this overexpression is partly due to more effective recognition and repair of DNA damages. However, the apoptosis frequency in larva and neuroblasts does not differ significantly. In the most cases, flies with the overexpression of this gene are more resistant to such factors as oxidative stress, hypothermia, or starvation. And the GAT45 is involved into longevity, possibly due to direct interaction with the DNA repair and the cell cycle proteins or RNAs. I would like to thank our sponsors, Science for Life Extension Foundation and Institute for Biology of Aging, and all my team. And thank you for your attention.
Very nice. So another gene, another protein involved in longevity. So we keep on building our potential targets for living forever.
Very interesting data, and especially I know it's very difficult to improve DNA repair because it's believed to be rocket capacity, and it's nice to see how to improve the DNA repair in that case.
All right.
But the recent year we published the science paper was SILT6 improving the non-homologous joining and homologous recombination, and it works through the PARP pathway, 6 PARP. I was wondering if there's any connection between GAT45 and this SILT6, because you have this nice scheme of the protein interaction.
Okay. Thank you for your comment. I think we can produce transgenic fly with your gene, and we could try to overexpress this gene and to see what happens with the longevity and aging speed. If you're interested in,
especially interesting because recently there was another paper published about SILT6 overexpression, and the effect on the lifespan was found only in the males mice, not in the females. And the same effect was
the same effect.
So it's a male. It's not clear why it's sex specific, but it's interesting.
I don't know why, but it's an interesting question.
Yeah, just a second.
Thanks for your talk. I have one particular question and one general question. The particular question is GAD45 only expressed in neurons normally?
I think normally GAT45 gene is not expressed. It's a stress response gene. Its level increased after stress for several minutes, and the half-life for this protein is very low. But we studied the overexpression just in nervous cells.
Do you have any plans to look tissue generally?
We have some problems now, but we are going to do this.
And a general question. Firstly, if you listen to this conference and many other, much of the literature, most of what we know about genes affecting aging is from knockdown studies or mutation studies. Your approach is to overexpress. Do you think if we were able to do screens using overexpression, our view of genes affecting longevity might be very different to the view that we have now?
It's a difficult question. I think my results are connected to the speech of Professor Ratan, because this gene is a stress response gene, and different kind of effects like heat shock or physical activity could affect the level of such stress response genes. But in young individuals, in old individuals, we couldn't, because they are suppressed. But we overexpressed this gene artificially. If we can affect the level of some stress response genes in old individuals artificially, it will be delay the aging speed. I hope so.
So, how much overexpression is needed to see the effect? 10%? 2%?
Since two times.
Yeah, but that's
And higher.
no, but
two times.
Is there technology actually
100.
available that we can do the titration?
I think we have to try to apply Alexander Stolzing technique to overexpress this gene in mice.
Also, my favorite question then, if you make a pill or something to do this GAD45, will it be anti-aging or is pro-health span? What do you want to do? You believe in
The problem
anti-aging
is
or?
that GAD45 effects in mammals depends on the tissue. In some cell types, the overexpression of GAD45 will induce apoptosis. In some cell types, it could induce stress resistance. That's why we can't apply just a pill for the whole body. We need to deliver, specifically deliver.
But as a general theoretical framework, again, are you working on anti-aging or pro-health span? I just keep asking that question to everybody.
I think pro-health
The remaining
span.
4 speakers also, please be careful.
I think pro-health span, but we hope that in the future we will produce approaches to delay the aging in human. Delay.
All right. Yes, aging.
So you said that GAD45 is overexpressed after stress response. So I think that it's really connected to homeopathic theory. So maybe at the low dose of exposure of the low stress response after exposure to low dose of stress, maybe GAD45 can be expressed and expressed. So
Yeah, but we observed that in old flies the level of GAD45 expression is drastically low, and maybe in mammals, I don't know, but maybe the promoter of this gene hypermethylated, because we know from the literature that in most types of tumors, the promoter of GAT45 gene hypermethylated. And maybe with the aging, we could see the same. And that's why we can't induce by stress in all the patients. I think so. I don't know, but
we cannot be more punctual than that. So we'll go for coffee, but please remember that first speaker after coffee break, Natalia Stefanova, she wants to speak in Russian. So those of you who don't understand Russian, please get your earphones or your translating equipment. So Natalia will be next, and then Guayjong Min, he will, and then we'll continue with Mikhail and Marina, hopefully. So enjoy your coffee now. Thank you very much.